I wanted to first start to identify the difference between two terms that are sometimes used interchangeably. The first term is heredity. And heredity is passing of physical and mental traits. This includes like behaviors such as talking with your hands. And again, they're being passed from one generation to the other. Genetics, on the other hand, is the mechanics of how these characteristics are being passed on, such as through DNA. I am going to introduce you to some new terms related to heredity. The first one is alleles. And alleles, I think of all the L's in allele, and these are letters. These are actually two letters. They symbolize one or more forms of a gene. So, for example, you're going to have one that comes from your mother and one that comes from your father. So there are two alleles. There are dominant alleles, as you can see here, and dominant alleles are represented by capital letters, and this is called homozygous dominant. Likewise, there are recessive alleles, and these are lowercase, and these are symbolized again by lowercase. So, for example, little b, one of these little b's came from your mother, the other one came from the father, and we call those homozygous recessive. Homozygous means same, and they're both lowercase, so they are recessive. For example, if B represents brown eyes and little b represents blue eyes, and again, the trait that we're talking about is eye color. So if it's big B, big P, you're going to have brown eyes. If it's big B, little b, you're going to have something called, that's also brown, but we call this heterozygous. Heterozygous means different, or it has a big B and a little b. It is different, and they are also both brown in color because they both have the dominant trait. Again, big B would be homozygous dominant. Big B, little b would be heterozygous. However, little b means they are blue and they are homozygous recessive. The last two terms I want to talk about are something called genotype. Gene, I think of the genes. They are the letters. We're talking about the alleles when we're talking about genotype. Whereas phenotype, I think of the pH or physical appearance, and that is the color. So that is the difference between genotype and phenotype. A Punnett Square is a diagnostic tool that agriculture, pharmacy, anybody who's breeding an organism might use. So for example, we're going to look at a monohybrid cross. Mono means one, so we're going to be looking at one trait. So we're going to be looking at an organism that has purebred brown eyes, for example, with an organism that has purebred blue eyes. So a Punnett square is basically a square that's set up like this. We have one parent, which is our purebred brown, goes on the top, and our other parent alleles go on the side. And it doesn't matter where they go. Again, one represents male and one represents female. So we're going to bring down and over a letter. So big B, or the dominant letters, always come down first. So we're going to have a big B. This one is a little b. This one's a big B, little b, big B, little b, big B, little b. So we're going to mix both those combinations. And again, the capital letter always comes first. We're going to cross our offspring from our previous cross. They're heterozygous. They're big B, little b with another big B, little b. So we set them up on our Punnett square. This is one parent here. This is our other parent. We're going to bring down this big B, bring over this big B. Again, remember the dominant always comes first, and then the recessive, big B, little b, little b, little b. So if we look at our colors, 3 of them are brown and 1 is blue. To look at these more closely, we have something called a genotypic ratio. Genes, or the letters. If I look for big B, I see 1. big B, heterozygous, I see 2, and little b, I see 1. Whereas the phenotypic ratio or phenotype is the appearance, the physical appearance. 3 of these are brown and 1 is blue.